would like to thank also the organizers of the module that invited me to give the talk. So as Antoine said, I work at the Wavefront Engineering Microscopy Group at the Vision Institute, which probably most of you know as the Valentina Emiliani group. But we do have a name. Other than Emiliani Lab. And indeed, we are a crowded group, so we are about 20 people, and we all work together with a common target to develop advanced optical methods for applications in studying neuronal activity. So the work here, I was told to show really the basics of our methods. So the slides that you will see in what follows is the work from current members, but also past members of the lab. So I will start with an introduction, why actually we need to develop advanced optical methods, as we call them. So basically, as I said, the goal is to understand brain function. So when you work in the interface with neurobiology, you want to satisfy the neuroscientists and the challenges that they have in their applications. And those challenges, they extend from a large spatial scale. So studying the brain means that you should be able to go through studies from the single cell level up to studying how single neurons interact within a neuronal circuit, for eventually understanding how circuits communicate between them. But also, you need to be able to perform studies in a relevant temporal scale. And this also goes from the millisecond time, or even sub-millisecond times, where you want to be able to characterize the precision of when an action potential is triggered, or even the frequency with which action potentials fire, for eventually to be able to reproduce the neuronal activity by replaying even asynchronous temporal patterns. So during the last years, advanced molecular developments have helped to this direction, and also the fact have helped also to the fact to the point to use light to perform those studies that traditionally were done by electrophysiology, so sticking electrodes to neurons. However, when you want to go to population studies, just using electrodes to go and study one neuron, it's not enough. So you need to be able to control populations of neurons. So nowadays we have the ways to express to our neurons molecules for recording their activity, fluorescent molecules for recording their activity, and also probes that help us to manipulate the neuronal activity, so to induce or to inhibit even neurons. And this is what we call the domain of all optical interrogation of neural circuits. And this is where the need for new optical methods that would enable to specifically control those tools became useful. So if we go first to see how we can record neuronal activity, well, this now it's done traditionally by using calcium sensors, and the most commonly used now is the sensors that belong to the GCAMP family. So in fact, before we were using dyes, now we use proteins because they can be easily genetically expressed to neurons, and they are less phototoxic, less invasive for the health of the neurons. And well, in calcium imaging, normally you had a talk on functional imaging at the beginning of Mifobio, so I hope you all followed that. Because yeah, a lot of I won't get into the details, I won't have the time, but briefly, these are sensors that proteins that they are tagged to GFP. And whenever there is an action potential fired by a neuron, there are voltage-gated dependent, calcium-dependent channels that are opening in the cell membrane. And so the increase in calcium changes the conformational state of the GFP protein, which gives the rise to increases of fluorescence of the sensor. Here you see an example on a nice example by Misha Ahrens on the zebrafish larva. And the thing is that this method is very sensitive. You can have a large signal to study the responses of the neurons, but it's not a direct measurement of the neuronal response. So in fact, you have the voltage change that induces the exchange of calcium ions of the neuron with the environment, and this leads to the increase of the fluorescence. So it's an indirect method to follow neuronal activity. But we also have voltage imaging sensors, which are, well, in the specific case, I have the example of voltage-sensitive domain sensors, which they are, as we will see later, most sensitive to two-photon excitation that we are going to use. And this is a more direct way to record neuronal activity because the GFP is tagged directly to the voltage-sensitive domain of the GFP, or the chromophore that's used is directly tagged to the voltage-sensitive domain at the cell membrane. And so any change of the voltage in the cell membrane is directly transformed to a change of the fluorescence in the fluorophore. Most of the cases, as you have seen probably by the talk of Michael and Lynn, we have a reduce in the fluorescence for these sensors, but now later developments also have inversed that, and we also have increases in the fluorescence. So that's why here you see a decrease in the signal. So the advantage of using voltage indicators, voltage sensors, for studying neuronal activity is that they are as I said, they give you direct response of the neuronal, a direct measurement of the neuronal response. So they are more sensible to single action potential detection, although now the latest GCAM sensors, they also do that, but they are able also to report sub-threshold activity, that is activity that doesn't forcibly lead to an action potential. And this is useful sometimes for neuroscience studies. The problem is that since they report action potentials last a few milliseconds, so you need to image them very fast to go up to kilohertz rates, while calcium sensors, they can be very well imaged even with a few hertz of acquisition rates. We will see later how this is a challenge. Now let's go to the photosensitive tools that we use for manipulating the neuron, so for inducing the activity to the neuron. Globally, those tools, they are divided in three categories. The cage neurotransmitters, that they are used for uncaging applications. You uncage the cage neurotransmitter with light. Then there are the natural photosensitive proteins and the small molecule photoswitches, which are basically engineered molecules, synthetic molecules that can activate neurons after light activation, illumination. And the advantage of this is that they can also be genetically expressed by the cells. So by a viral injection, you can in your preparation. Well, in our case, we mostly use mice. You can express both the activity sensor and the optogenetic actuator. And like that, you can have the full control. So how here we will focus mostly on the natural photosensitive proteins on this category, which are proteins that we can find in nature, basically in a specific type of algae. And the property that they have is that they are basically ion channels that they are expressed on the cell membrane, and once they are illuminated with light, they change their conformational state. And the channel, as it absorbs light, it opens and it leaves ions going through the cell membrane. So back in 2005, researchers managed to express that kind of proteins to mammalian neurons. And the result was quite spectacular, because in fact, upon expression of this kind of proteins on the neuronal membrane, by illuminating with light, they were able to induce activity, and by just controlling your light illumination, you can control the activity and the spiking frequency rate of the neurons. So that marked the beginning of optogenetics. And now it's been almost 20 years, and it really dominates neuroscience. But it was not, so it dominates neuroscience not without a reason. Actually, nowadays there are proteins that they can be excited at different wavelengths, or even proteins that inhibit neurons instead of exciting them. And actually, there are hundreds of channel rhodopsins today, which basically they differ in the variety in what concerns the photocurrent size that can produce in neurons, the ion selectivity, whether they permit sodium or other types of ions to go through the membrane. Of course, they are activated in different wavelengths, and this is also useful, or they have different kind of kinetics and light sensitivity. So depending on the application, neuroscientists choose the proper tool to work with. And what are the illumination methods that are used, were mostly used at the beginning when optogenetics has came onto the stage, where basically the first studies were using just an optical fiber that was delivering light unselectively to neurons. So to the preparation in this case is on mice, and like that all the neurons were excited at the field of view, and the selection was based only on the selectivity of the expression. So normally the expression, the virus that you use to express has a specific promoter that targets specific types of neurons. And these kind of fibers, they were even combined with electrodes, such as to excite the neurons and to record the neuronal activity at the same time. And there is really a lot of studies that have been done at the beginning of the genetics using these tools. So they were really, it was really a landmark in neuroscience. However, there were still a number of studies that they could not be done because of the non-selective illumination. So, for instance, if we want to perform studies concerning functional connectivity, or establishing a critical number of neurons that we need to excite to control a neuronal network, or even if we want to produce asynchronous patterns, when, as I told you at the beginning, we need to be able to excite specific neurons with a good lateral and temporal resolution, and if possible, simultaneously. So to control the number of neurons that we excite simultaneously. So we need for more sophisticated optical methods. And so the challenges, as I said, is we need good spatial resolution. We need to be robust to scattering, to be able to excite even in 3 dimensions, because the brain is 3-dimensional, to have temporal precision of the order of millisecond, and to be able to excite and record all optically. So for the first 2 challenges, in our lab, we have chosen to work by using 2-photon excitation. And in two-photon excitation, basically we have two approaches. We can either serially scan a fast spot around our field of view or specific areas in our field of view. Or we can use parallel illumination by illuminating the sample with specific light-shaped patterns. So I won't stay too much here. So I guess all of you know what two-photon excitation is about. So basically, the interest of using two-photon excitation is that we can have optical sectioning that we don't have if we use visible light to excite our preparations. So in that case, we have a lot of autofocus light that destroys our spatial resolution. And the second advantage is that in two-photon excitation, we use infrared wavelengths that help us to go deeper and to study neurons that they are more deep in brain layers. So starting by the scanning approaches, the idea here, I guess you all know, is that we use a scanner that is placed at a plane that is conjugated to the back focal plane of the objective of our microscope. And this scanner can be whatever, can be galvanometric scanners, resonance scanners, or acousto-optic deflectors. And the idea is that like that, we scan the laser beam at the sample plane, and we can image a whole field of view, or as I said, we can perform scanning of smaller areas, ROIs in our sample. It depends on the application. Well, particularly for using this for optogenetics, the problem is that the scanning methods are relatively slow comparing to the time scale of physiological responses and reactions of neurons. So basically, we are limited by the dwell time that we need to stay on specific location to induce a physiological response. The scanning time depends on the time to move from one side to the other, depending on what we want to scan, if we want to scan a whole field of view or specific neurons. And all this has to be multiplied by the number of sites. And if you want to do this for many neurons, you have to multiply also by the number of neurons. So basically, particularly for optogenetics, the people managed to use scanning methods by applying particular trajectory types. That was the only way to have the necessary dwell time and to scan as fast as possible to have an activation of the neuron, to be able to control the neuron. So ideally, we would need to increase the illumination surface, such as at least to save the time that we spend on scanning the beam from one side to the other. So that's what we propose in our case. Basically, especially if you go to two-photon excitation, the volume that you excite with your activation beam on the cell is very small, much smaller also in Z, because you have optical sectioning, and like that it's impossible to generate cellular responses if you don't scan fast your beam all over the cell. So how do we increase the excitation area without sacrificing time? Basically by using parallel illumination methods, and this can be by using either amplitude modulation or phase modulation. In the amplitude modulation, basically you put this time it's not a scanner, it's a machine that modulates your beam. A special line modulator is placed at a plane that is conjugated to the image plane of your microscope. Like that, supposing that we have a preparation where we want to illuminate a certain region of interest. In the case of amplitude modulation, we can directly address the form, the shape of the beam we want to create. For instance, by controlling if our SLM is a a digital micromirror device, we can control which mirrors we turn on, which mirrors we turn off. And like that, we have directly our shape at the focal plane. In case of phase modulation, what we need to do is basically our spatial light modulator is conjugated to the back focal plane of the objective. So here we are in a Fourier plane, and what we need to do is to produce the Fourier transform of this pattern, such as after going through our lenses to give the desired intensity. So in this case, we have some computational time that we have to take into account. But in any case, now there are SLMs that can go reasonably fast for performing this kind of activation. The advantage here, as I said, is that we don't depend on any scanning time, but we have just the dual time that we need to depends on biology clearly, and it's the time that we need to induce a physiological response. So in our lab, we use particularly phase modulation, because with amplitude modulation, the problem is that you lose, in fact, the power that you don't send to your specific targets. And for those, I guess most of you are physicists here. So basically, for phase modulation, we use devices that take advantage of the local change of the refractive index of the pixels. And so this is done by using liquid crystal molecules controlled by a certain voltage. Basically, by changing the voltage at each pixel, we change the orientation of the liquid crystals, and this changes the refractive index, and by changing the refractive index, we change the velocity with which each, let's say, each ray is reflected from our device. By doing this for all the pixels of our device, of the SLM, at the end, we have an arbitrary modulation of the phase wavefront of our beam that can give rise to different excitation patterns, either multiple diffraction limited spots or really arbitrary patterns that can be chosen on the base of our desire, which areas we want to excite at the sample. And basically, because by doing this method, because we just do everything computationally, the method, we call it computer generated holography, because it's based actually in the classic holography, only that this time we don't have a real object here. We just replace that by the virtual object that we want to create at our sample. And then the only thing we do is to calculate the phase profile, the Fourier transform that we give this when we will do it inversely. And so in that case, we don't have an object, we don't have an illumination beam that goes through the object, but we just have a reference beam that we send to the SLM that is addressed with this phase pattern, this Fourier transform. And then when the SLM is illuminated with light, it gives us back the hologram, which is the desired pattern that we want to have at the sample plane. Briefly, this is just having in an optical microscope. This is just having an SLM at the back focal plane of the objective. But because the SLMs, I guess you have seen here in the school, they are not just small elements that you can put at the back aperture of the objective. Usually we put it, as I showed you in the previous scheme, with a telescope conjugated to the back focal plane of the objective. And to generate these arbitrary excitation patterns, you can use iterative Fourier transform algorithms of the type of Gerberg and Saxton, for instance, to perform back and forward Fourier transforms until you get, after a few iterations, you can get the desired excitation intensity pattern. If you just want to project diffraction limited spots, there are simpler algorithms that you can use, where you just have to encode your SLM with a grating phase pattern or with a lens phase pattern, or with combinations of both if you want to project spots both in XY or in Z. And these are some examples of patterns that we have generated over all those years in the lab. So you can you can really match your excitation to the form of the neuron that you wish. Or even project, I don't know if you can see with the light, project patterns in 3 dimensions, or even generate sequential projection of phase patterns and generate like that dynamic holograms. Of course, the speed of the refresh rate of SLMs is still limited in relation to other methods that you have eventually seen, like acoustic deflectors. But everything depends on the application you have. So in most of the cases, this is enough. And in the lab, we have also proposed methods to improve this speed, in which but I won't refer to that because I don't think I will have the time. So the advantage of these holographic beams is that because we do phase modulation, we always illuminate the back aperture of the objective in a steady way. So this means that we can always feel the back aperture of the objective. So comparing, if we wanted to generate the same extended pattern, let's say of, usually for neurons, we do 10 to 15 micrometers of spots with diameter 10 to 15 micrometers. So if you wanted to generate a similar Gaussian beam, actually you would need to underfill the back aperture of the objective, and so you would lose the axial confinement. You wouldn't use the full focusing power of your objective. You would lose in terms of axial resolution. With computer-generated holography, we can always keep a nice axial confinement because we always illuminate the whole back aperture. The light is redirected to the places we want just because we do phase modulation, okay? But still, when we use 2-photon excitation, things get even better because we have the 2-photon effect that improves our depth of focus, if you wish. However, the problem remains if we start to illuminate a lot of targets, and it becomes even more severe if the targets, they are close together. Some years ago, my main goal in the lab was to find for a solution to this problem. And so we thought of using the technique of temporal focusing, which is in fact it was introduced in nonlinear microscopy as a way to get rid of the out-of-focus light of large illumination areas. So we said, what about if we combine this with holography? So a few words about how temporal focusing works so that you are able to follow better the next results. So basically in temporal focusing, what we do is that we take advantage of the fact that the 2-photon excitation signal is inversely proportional not only to the size of the excitation beam. So this is the reason why we focus our beam sharply when we want to do 2-photon microscopy. But also, it depends on the pulse duration. So in temporal focusing, what we do is instead of focusing our beam very sharply, because we don't want that, I remind you that especially for optogenetics, we want to increase the illumination area so that we recruit most of the channels simultaneously. So what we do is we play with the pulse duration of our femtosecond laser. So we do such as we increase the pulse duration out of the focal plane, and we keep the original short pulse duration in the vicinity of the focal plane, and so the 2-photon absorption becomes less probable out of focus. How do we do that experimentally? Basically, we use standard techniques from ultra-fast optics, and we use dispersive grating to disperse the spectral frequencies that they are contained in a femtosecond pulse. And with the help of a telescope, which is the lens and the objective, we refocus our spectral frequencies at the sample plane. But because we introduce, let's say, a different optical path for each color, for each spectral frequency, at the end, these frequencies, they don't recombine in phase in the whole focal volume. So actually, out of the focal plane, they recombine with a certain phase delay, and this gives rise to a broadened laser pulse. In other words, we manage like that to have short pulses only around the focal plane. So, in fact, the frequencies arrive in phase only in the vicinity of the focal plane. And in this way, the fluorescence is basically excited only around this volume. And if you design your temporal focusing system well, meaning that you choose the right dispersion for the grating and the right focal length for basically for this lens, because this is the objective that you use for your application, you can manage to have reasonably good axial resolutions of the same as if you were doing a line scanning microscopy. Because in fact, I can show you afterwards that temporal focusing at the end is like performing line scanning to photon microscopy. Okay, so let's see the results. So here's the previous slide when we had a lot of holographic spots placed together, but combined this time with temporal focusing. So you can see that the autofocus light is rejected, and the axial resolution actually gets to the same levels as for a single spot. And what about three-dimensional excitation with temporal focusing? Well, in that case, we have a problem because when we introduce temporal focusing, we have the grating is based at the image plane of our SLM. So basically here we have the shape of our computer-generated holography method. And because the grating is just a plane, we cannot project to the grating 3D patterns. So basically, if we do temporal focusing, the different beams, the different shapes will be all projected to the same plane. Otherwise, we don't see them at the focal plane. Their fluorescence is suppressed. So to manage to separate them again at the sample plane, we basically need to introduce another phase modulation, but already at the Fourier plane where the frequencies are separated, and like that we can apply a less effect on the second SLM for each pattern, and thus separate again the shapes in the axial direction. In this way, we managed to show that we can actually have several temporal focused spots in a volume, and they can be all nicely confined without having crosstalk in the excitation between them, even when they are fairly close together. And this basically we can do it more simply by having our first SLM projecting a pattern and the second SLM performing basically a cloud holography, so just diffraction limited spots. And this first spot is then multiplexed in the different positions that they are encoded by the second SLM. So as I said, these techniques, we use them mostly to perform all optical studies and optogenetics, so activating different opsins in neurons, in brain slices or in vivo. Here is an all optical experiment in vivo where we control a number of neurons, and in red they are those that they are excited, and in yellow here, and in white here there are those that they are not excited. And here is an example where we can really be very specific by using this kind of illumination. So if you separately illuminate dendrites or axons, or the whole neuron with its dendrited axons, you can really tune the size of the neuronal response. And recently, we have also shown for applications that they need to be on freely moving animals. We have shown that this technique actually works, so holography with temporal focusing actually works nicely also through graded index lenses. And when we combined these lenses with fiber bundles, we are able to finally perform experiments, all optical manipulation of neurons experiments, also in freely moving mice. So here's an example where there are specific neurons in the field of view that were illuminated, and here we see that only the neurons that are illuminated are responding to the stimulation. So basically, here we combined we were scanning a large spot through the fiber to do the calcium imaging, and we were activating selectively the neurons with computer-generated holography. You can have more details on this on the paper, or I can give it to you later. So what about functional imaging with our techniques? So of course we are not the first that have thought to use computer-generated holography for doing functional imaging. And there are also papers, for instance, from the lab of Tommaso Fellini in Genova, where they use this for doing, they use holographic spots to show that scanless calcium imaging can be actually better than the standard scanning calcium imaging with galvanometric mirrors. Well, the problem is that when you go now to voltage imaging, as I told you at the beginning, the voltage sensors, they are really confined on the cell membrane because they really attach to the voltage sensitive domain. And so we have very few molecules. It's not like the calcium sensors that they are expressed in the whole soma. And so you have bright neurons that you can see and you can excite them and have a large signal to noise ratio. So first, by being like that, we risk easily to photo damage our neurons, to be phototoxic. If we want to send a lot of light to have a high signal to noise ratio, there are very few photons that can be absorbed by this thin membrane of the neuron. So we need to illuminate with high intensities, which can destroy our neurons. And we need to go very fast because the voltage sensors, they report action potentials that, as I told you, they are of the order of 2 to 3 milliseconds. Recently, with the development of voltage, of genetically encoded voltage indicators, it's much easier to perform this kind of studies. And in general, because before with voltage sensitive dyes, the experiments were almost impossible. But now these sensors, they are genetically encoded as well as like the calcium sensors. And so people have started using the easiest way to go and image them, which is to use a wide field illumination in a microscope. This is good because it gives you long dual times. And with wide field illumination, you illuminate your whole field of view, so you have high acquisition rates. And you have a good lateral resolution because you use visible light, and the implementation is pretty simple. It's just a wide field epifluorescence microscope, but you lack optical sectioning and you have a lot of out of focus light. So of course then the next idea is to use targeted light instead of wide field. And we have also shown that computer-generated holography in single photon excitation works as well. But this is a nice example from Adam Cohen's lab in MIT, where they use digital micromirror devices to do targeted illumination. And like that, they managed to do nice experiments without having the problem of the out-of-focus light. Nevertheless, this remains a single photon excitation, so it means you are limited to the depth, and it's also, as you can see, it's camera detection. So Nevertheless, they managed to go up to 300 micrometers. But their method was based on sparse labeling strategies to avoid to have out of focus excitation. So people started to think ways of using two-photon excitation, but going really fast up to kilohertz rates in order to be able to image voltage sensors. So we had during the last years the development of really nice optical methods, like by the group of Stéphane Dieudonné, where they used the microscope with acousto-optic deflectors to perform voltage imaging, because with AODs you can really scan your sample at very fast rates. Nevertheless, they were obliged to scan not diffraction limited spots, but to create mostly patches of, let's say, of illumination, so that they take, so that they are not limited by the movement of the mice, and also to be able to record more signal from more molecules. Then there were this nice method by the group of Nagy, but it's really particular. You should use really optical developments that they are really specific. And lately we had also a microscope based on spatiotemporal multiplexing combined with deep learning methods to achieve to retract the signal from depth. So in our case, we were asked whether we could use, since for doing voltage imaging, we want to go fast. So we said, why not trying, since we are scanless, to see if we can image voltage with parallel methods. And we used the recently developed molecule JEDI2P, which is particularly optimized for two-photon excitation. And we tested in organotypic slices first, where we had a really dense expression. And you can see we could nicely resolve single action potentials by, this is an experiment on single neurons, and you see how you can really target a single neuron within this mess. So we had practically the same delta F over F and signal to noise ratio as the previous two-photon excitation methods that I showed you. We were able to also image a neuronal activity in different frequencies and for acquisition rates down to 500 Hertz. And also we managed to image spontaneous activity. This is the electrophysiology recording, and this is the optical recording. And you see how good they fit. And even under some circumstances, for instance, by averaging several trials, we could image sub-threshold activity with JEDI, which is not a sensor optimized normally for sub-threshold activity. And we could image down to changes of 1 millivolt on the cell membrane. And then, because we wanted to test if we could also perform multi-cell illumination, well, I forgot to mention that the JEDI sensor normally has its peak at 940 nanometers, but with the power we were needed for imaging a single cell with standard laser oscillators at 940, we could not get enough cells. So we tried to use a laser that a low repetition rate with higher pulse energy that we had already in the lab, but that was at 1030 nanometers. So we tested the sensor at 1030, and we saw actually that even though we had we were losing about 40% in the signal to noise ratio just by shifting the wavelength out of the peak, we could still image nicely the cells. This is in vitro spontaneous activity, so unfortunately we were stimulating all the cells, but not all the cells were responding. This is a particularity of the neurons on organotypic slices. And then we also performed experiments in vivo we went down to 250 micrometers, and all this is camera detection. There's no PMT detection here because we illuminate in parallel. And we were able again to have a nice signal in the cases where the neurons were responding. These are experiments in the barrel cortex. So this is promising for the future. And now I will switch to a last example where we used our techniques in showing you some experiments for the study of functional connectivity in the mouse retina. So basically, the mouse retina, this is a nice application. The retina in general, okay, it's a layered structure that in fact, its role is to record visual scenes and to transform them through a really diverse optical pathway. The information is received by the photoreceptors. Then it's treated by the bipolar cells and transformed vertically to the ganglion cells and horizontally through the amacrine cells. So at the end, we have the retina transforms the input image into different scenes, into different representations, that they are all encoded in a different part of our brain. And when they are combined together, they give the final result of the visual perception. So what we wanted to see, in fact, an interesting question for neuroscientists that they deal with the vision restoration and the retina diseases, is how the information is transformed from the bipolar cell layer to the ganglion cell layer. So what we did in the lab, in collaboration with a group of Serge Picot and Olivier Mah at the Vision Institute, was to express an opsin at the bipolar cell layer and the calcium indicator at the ganglion cell layer. And with the holographic microscope and the holographic methods, we were imaging the calcium cell, the ganglion cell layer. That was regular, standard scanning imaging. But with a holographic microscope, we were selectively activating the bipolar cells in different group ensembles, and we perform a study on the functional connectivity between this cell layer and the ganglion cell. So we were basically studying by activating specific neurons here, which ganglion cells are connected to these ones. And we managed to get some results to get an idea how this story works. And where we could apply this thing. Actually, in the Vision Institute, there is a very recent study where there has been the first optogenetic application in a clinical level. And they expressed an opsin at the ganglion cells of a human, okay, where he suffered from retinal degenerative disease, retinitis pigmentosa, which can gradually lead, in fact, it's a disease where the photoreceptors are degenerated gradually with the age, and it can lead to blindness. So they expressed an opsin, as I said, to the ganglion cells, and they activated the ganglion cells with a set of goggles. So the patient was wearing goggles that they were transforming with a camera that was sensitive to intensity changes, the object that they were showing to the patient. It was transforming this to a light signal that was illuminated the ganglion cells where the opsin was expressed. And they showed that actually the patient was able to distinguish objects, and to say, for instance, how to number the objects or to perform such basic tasks. For instance, to say which object was bigger or smaller. This is a nice application where actually our holographic methods could be applied, and this is something that we target to. Basically, as in that case, the optogenetic activation of the ganglion cells was really unspecific. So the camera was recording the object, it was transforming that to an intensity pattern that was sent to the patient's eye. The goal is to be, if we want to be more specific, for instance, in this way, the patient could never be able to read something, to distinguish letters, because this is a detailed pattern. So what we tend to do in the lab is by using projection of more specific patterns to see which are the ganglion cells that are responding to specific patterns. And then by using SLM targeted illumination through the goggles to retarget the same ganglion cells and see if the patient can perceive the same information as he would do if he had a healthy retina. So this is a nice application where we hope to apply optogenetics for vision restoration with specific holographic illumination. So to summarize, because I think I'm just over my time, so I showed you that today there is a growing molecular development where it gives us sensitive sensors and actuators to use with advanced optical methods for the neuronal, for the all optical study of the brain, of neural circuits. With advanced optical methods such as computer-generated holography with temporal focusing, we can have great potential to apply to this kind of studies. Holographic functional imaging can also be possible even with voltage indicators, not only with calcium. And holographic manipulation of neurons can have a wide resonance and applications in several fields of neuroscience that I haven't shown you here, but you can do studies concerning perception. These are studies that they are already published, pathologies, or as I showed you here, for vision restoration in the retina. So this is again, as I said, a collaborative work. A lot of people have worked for that. Also, a lot of people, this is done with a lot of effort to get financing from different sources. And I will take advantage before I close to advertise two events. One is a training course that we organize every year in the lab. So if you are thinking that these kind of methods could be useful for your project, for your PhD, for your postdoc, just write us, and this may be interesting for you. And then a second event, well, if your domain is to use light for studying the brain, this is the conference to be next June in Paris. And thank you for your attention.
Thank you. My question is about power. Where you illuminate several neurons, at the limit, a big surface. In addition, you use a CCD to detect not a photomultiplier, which I suppose still remains less sensitive than
for the case of voltage imaging. Yeah.
Yes, voltage imaging. Yes, so I should have said it. So
yes.
How much power do you need, or to put it otherwise, how many neurons can you image or stimulate?
Okay, so in the very first experiments we did, experiments in brain slices, and we had, as I said, a standard laser oscillator, 80 megahertz laser. So to image one neuron, we needed something like from 75 to 125 milliwatts per cell, which, if you compare with other scanning methods that they use from 20 to 30 milliwatts per spot, is a lot. But the thing is, first of all, on light intensity, we are lower because we illuminate a large surface, so we don't have linear photo damage. And second, when we switched to a more energetic laser, so with more energy in the light pulses, we could reduce the power down to 5 milliwatts per cell for imaging one cell. And that was even in vivo in 240, 250 micrometers. We needed something like 7.5, 7, 8 milliwatts per cell. So that brings us back in a good shape. But still, if you want to increase the number of cells, as you said, you need to increase your power. And then you get back to the same result. And for sure, we hit. That's for sure. The more you increase the average power, the more you hit. So basically, there has to be, but this is for all methods using lasers, there has to be an effort. There is a limit of the maximum number of cells you can do for being in the safe, let's say, heat photodamage level. And by simulations, we see that in our case, with these parameters, we are at about 10 to 15 cells. Nevertheless, it's not always like that in the literature so far, but that should be the tendency if you want to be safe, that you don't induce something to your preparation, to your tissue. But this depends also on the sensor. There are a lot of parameters that you need to take into account.